So thank you, everyone. Welcome back from lunch. Our next session, session 4, is on experimental models and functional approaches. And given the diversity of approaches, we have 2 applications speakers in this session. But first, we have Dr. Stephen Montgomery from Stanford University, who will be setting the stage for this discussion, and he'll also be serving as moderator for this session. So please take it over, Dr. Montgomery.
Thank you very much, Lee. So to start off, I just wanted to give a really high-level overview to some of the topics that we'll be discussing in session 4 today. And again, as mentioned, we're focusing largely on experimental models and functional approaches that can support understanding of gene by environment effects, particularly as they relate to cancer. So I think many of us are aware of the general problem is that we have observations at baseline of disease incidence, but then with respect to an environmental perturbation, we might see an increased observation of the incidence of disease. However, there are several challenges with doing this type of work. The first being imperfect measures of exposure. So in many cases, we might not know exactly what environments are underlying this increased incidence. And it might also be a multitude of environments. This is something that's come up earlier. The second is that there is variability in natural populations. So whether or not it's genetics or a range of different additional exposures or confounders, these can also influence the types of, our types of confounding when doing this analysis. There's also limited molecular understanding. And so this gets to one central feature of what this session is going to explore is that when we consider, how the environment might increase risk of disease in epidemiological setting, we don't actually understand what are the molecular pathways or pathological cell types which might fundamentally be involved in the etiology of disease. And then it's also very hard to prove causality. So without using, in natural human populations, people don't typically like being poked or prodded in different ways to see how it might impact disease. And so a lot of analyses needs to be retrospective. However, if we think about model systems and different functional approaches, we have the opportunity to actually introduce an intervention and understand the causal relationship of that environment on some cellular phenotype that might be relevant to that disease. So there are lots of opportunities for boiling down what are population studies into different model systems and exploring a range of different omic traits. And we're going to hear about some of those today. But really, trying to figure out what are the best model systems and what are the best omics traits remains a fundamental challenge. And this schematic here is just representing the range of different molecular assays. Again, not entirely complete, that are generally used to explore cellular function. And these can be from assays which explore DNA accessibility and transcription factor binding, to methylation assays up through transcription and translation to now protein quantification, which can be quantified both with mass spec and now sequencing-based technologies. So one of the benefits to thinking about these types of model systems, and particularly exploring different omics level phenotypes, is there's the opportunity to actually increase our ability to measure different effects, to reduce experimental or confounders that might exist in the natural population. And so to think about how we can find from what might be a very noisy signal to what might be a more robust signal in terms of that environmental perturbation. There's also an opportunity, and we're seeing increasingly studies over the last decade that have aimed to explore gene by environment effects by considering what's happening at the omics level instead of large-scale population cohorts. And so one just quick example of this is you can imagine having an omics trait from a population sample at large and looking to see how a particular trait actually stratifies by genotype or genetic information. You might see that there's actually no difference whatsoever. Whereas in the context of the environment, you can actually see that genotype actually matters for stratifying that omics trait of interest. And so this can be very relevant if in your disease context, you also see that there's stratification of incidence in the same way with respect to genotype, because now you have some kind of molecular basis for what might be driving that underlying increased risk that would be missed just by studying it at baseline. So we're going to talk a little bit in the rest of the session about different experimental models of environmental exposures. And these are going to boil down into understanding in vitro models, whether or not they're cellular, tissue, cell line based, to organoids, which are becoming a very interesting and well-used model to explore more complex tissue environments, and also preclinical models, which have well served the field in many different cases. So just to give you one example of where these types of studies have benefited, these are early studies that have considered primary macrophages, which have been cultured across a range of different strains, but can also be exposed to a range of different environmental perturbations, whether or not it's LPS or oxidized lipids in this case. And through these types of assays, you actually get genetic information, you get that controlled perturbation, but you can leverage the genetic variability in these models to actually look for gene by environment effects and how they might ultimately relate to differences in immune response. You can see these types of effects, as I was discussing earlier, where in the context of different perturbations, so here this is tuberculosis infected dendritic cell lines, and in the context of the non-infected dendritic cell line, you can see there's a genotype stratified effect. However, in the infected state, there's nothing. This is just showing the opposite here. So you would miss these types of genetic effects if you weren't looking at these perturbations in culture in these contexts. And it can actually relate to how people might respond to disease or treatment in those particular exposures. There are several challenges with these types of G/E studies as they relate to doing cellular studies with model exposures. And so we'll talk a little bit about these as well. There are many environments to test, so it's expensive to imagine collecting cell lines from numerous people or numerous animals, perturbing them all, and then understanding how genetics responds to those perturbations. It's also a challenge that there are limited numbers of cell types tested. So in many cases here, we're looking at cell lines and we're perturbing them with one type of environment. And so this is where organoid models are showing a range of promise, particularly in the context of cancer. So it allows us to profile a range of cellular phenotypes, but then also a range of cell types through developmental trajectories. So this is one example from our exceptional review that I encourage people to take a look at, which actually describes the range of different organoid model systems that have been developed with respect to cancer. So largely looking at normal organoids and how they transition to tumor organoids through exposures of different microorganisms, environmental chemicals, dietary factors, alcohol, in this case it's ethanol, stimulation on the cell lines, or various aging related pathways. So there has been established literature exploring this in great detail. However, one challenge that still remains with organoid models is that it's still difficult to study G/E. So in terms of collecting the same number of cell lines, profiling genetics, and doing those types of perturbation assays that are more commonly done in cell lines, there haven't been any, if many studies have done that to date. So there's also an advantage in organoids, and we're seeing this more and more in terms of getting patient-derived organoid models. So instead of thinking about reference model organoids, actually getting these directly from individuals who might be undergoing some kind of cancer treatment or disease incidents, and then actually exploring how they respond to different treatments. And so one, I think, impressive study was in Science a few years back, where they actually were able to get patient-derived organoids from metastatic, heavily pretreated cancer patients recruited in phase 1 and 2 clinical trials. And they were able to show that for these patient-derived organoids, they could analyze therapies outside of the context of the patient. And they were very effective in predicting the ultimate response to different agents and chemotherapy. So we can imagine how these types of models might exist side by side with different individuals in the future. And then finally in this session, we're going to touch on different preclinical models as well. Spend most of our time talking about zebrafish as a model. This is just one picture from a recent review showing the range of different cancers that can be explored across a zebrafish. These allow you to prospectively study very complex exposures, so you can consider not only single types of perturbations, but combinations of perturbations. You can also access, hard to acquire cell types and tissues through these types of systems and trial different therapeutic interventions. The challenge is, though, and I'm sure many of you are aware, that very few preclinical model studies actually translate to humans. And so we have, these exciting systems, but how we can actually think about what types of data move the discoveries we're making in these model systems to humans remains a significant challenge. So in general, this session is going to cover the power and challenges of using cell lines and patient-derived tissue cultures. It's going to highlight where organoid research can benefit, and it's going to discuss some of the strengths and limitations of animal models for cancer, for studying tumor genesis and evolution, including using zebrafish. And we're going to highlight where novel omic strategies can expose environments and different gene by environment effects. So with that, I'll conclude the introduction, and I will move us on to the first talk. Just give me 1 second to transition here. Okay, so we have two speakers that are going to start us off in this session. The first speaker is William Hill, who's from the Francis Crick Institute.
Hi, thank you for the opportunity to speak today at this really interesting workshop discussing the environmental data and other omics. And so today I'm going to talk to you about our study exploring EGFR mutant adenocarcinoma and the promotion by air pollutants. And we're studying lung cancer in people who specifically had never smoked, because it's a very distinct form of the disease from smoking lung cancer. It's got a low mutational burden. The most common altered driver gene is EGFR, and there are these clinical disparities that it appears to be more common in females and in certain parts of the world. There's a number of established risk factors which may explore this, such as germline susceptibility, radon, secondhand smoke, and exposures. And it appears that potentially the proportion of people who never smoke who get this disease is on the increase. So there's an unmet clinical need to understand the biological underpinnings of this patient group. And what's recently been found by whole genome sequencing of these tumors is that the vast majority of mutations that arise within these tumors do not appear to be in the context of an exogenous mutational signature. Around 97% of over 200 tumors where they carried out whole genome sequencing displayed an endogenous mutational signature indicated in gray here, perhaps suggesting that carcinogens are acting in a non-mutagenic fashion. And so this, I think, brings us to the question of the models by which tumors can arise. And it's clear that factors such as UV light and tobacco smoking induce mutations. And it logically follows that the more oncogenic driver mutations, second hits that arise, the increased likelihood that you have of tumor growth. But there's a number of additional considerations for these models. For example, the BRAF V600E melanoma is not actually within the UV mutational signature. And a number of increasing studies are showing that normal healthy tissue harbors mutant clones that are supposedly oncogenic driver mutations, but there's no clear evidence of cancer. And more recently, exposure of mouse models to 17 out of 20 environmental carcinogens were able to promote tumor genesis without increasing DNA mutations or specific signatures. And even in our cohort of TRACERx lung cancer patients, in people who do smoke, it does appear that around 10% of people do not have driver mutations induced by the tobacco smoking. So this, I think, leads to a possible additional or alternative model to tumor promotion, which was originally proposed back in 1947 by Berenblum and Schubrich. And it kind of brings up this idea of a two-step model whereby you have an initiator, which was induced by DMBA, which was later found to induce the mutation, and then a secondary hit, which they found as TPA as an inflammatory, which induced these pre-existing mutations to drive clonal expansion and selection of these mutant clones and drive towards cancer. And so we wondered whether lung cancer in people who had never smoked, which is known to be associated with levels of air pollution, might be acting via this non-mutagenic mechanism, and whether this opens up opportunities for therapeutic intervention or identifying those at high risk. And then just to briefly introduce PM2.5, so these are air pollutant particulate matter, which are smaller than 2.5 microns. And here's just a graphical distribution of a subway station in the UK, showing that the chemical composition can change through the day, and also the amount can change through the day. And it can be a very localized change compared to the background London mean, and the various components that can be made of this. And so to study this, we took a three-pronged approach, carrying out ecological correlation analyses between EGFR mutant lung cancer and PM2.5 pollution levels, and developed mouse models combining genetically engineered mouse models with exposure protocols to understand the mechanism. And then finally, we used multiple normal tissue cohorts to understand whether these oncogenic EGFR driver mutations are present within normal lung tissue to fulfill the two-step promoter model. And so just initially, we carried out ecological correlation analyses within working with Public Health England and DEFRA within the UK, looking at the regional incidence of EGFR mutant lung cancer per 100,000 and the mean PM2.5 levels. And we found a significant positive correlation between these, possibly suggesting that we might be onto the right track and that there is an association. And then we further explored this in two other in-country cohorts, both in South Korea and Taiwan, which have increasing levels of PM2.5 compared to the UK. And again, we saw still this positive, significant association was retained. And so then moving into the mouse model, so then we went to explore whether this was a causative and functional link. And so what we did was we used 3 genetically engineered mouse models where we induce expression of the oncogene at day 0, and then we give intratracheal administration of particulate matter for 3 weeks at physiological levels. And then we're assessing tumor burden at extended time points in these mice. And what we found is in these 3 different mouse models, in the first on the left, we found that these early neoplastic lesions that arise by 10 weeks post-induction were significantly elevated in mice exposed to, in a dose-dependent manner, in mice exposed to particulate matter. In a more aggressive CCSP model in the middle, we found that there was a significant increase in the actual adenocarcinoma that formed within these mice. And finally, we wanted to test whether it was specific to EGFR or whether KRAS was also impacted. And so we used the classic JAX model of lock, stop, locks, KRAS G12D. And again, we saw that there was a significant increase in the number of lesions that were developing. So this suggests that we can model this interaction in the lab, and we can use this model to understand the underlying biological mechanism. And so we first set out to test whether is PM acting as a mutagenic factor in this model, or perhaps in a tumor promoting model. And so we carried out whole genome sequencing to 100x of tumors within these mice. And we saw no significant increase in the mutational burden within tumors of mice exposed to particulate matter compared to controls. We saw no significant increase in any specific trinucleotide context or specific base change. And when we looked at the signature profile, we saw there was no difference in the mutational signatures that were coming up. So this does suggest that in our model, exposure to particulate matter is not elevating the mutational burden. So perhaps it's acting in an alternative fashion. And so what we did was we crossed our genetically engineered mice to Ragnall IL-2 receptor gamma null mice. So these mice are broadly immune deficient. They're lacking T cells, B cells, NK cells, and also a disrupted myeloid compartment. And when we repeated the same experiment, the same exposure protocol and harvesting, we saw no significant difference in the amount of tumor burden within these mice. So this does suggest that a competent immune system is required to mediate this particulate matter promotion effect on tumorigenesis within our model. And so we carried out a number of different analyses to explore which immune compartment is being altered by particulate matter. And I'm just showing the IF here for the sake of time. But what we identified was that macrophages seem to be a key responder to particulate matter within our system. We found that directly after finishing the exposure protocol, there was an elevated number of macrophages within the system. And even at 10 weeks post-oncogene inductions, this is almost 2 months past finishing the exposure protocol, there's still elevated macrophages, perhaps suggesting some sort of feed-forward loop between the tumor that's arising and the macrophage response. And I think one of the important things within this study is that what you find in a mouse can allow you to understand what you're finding in human patients. And so what we did was we took normal samples from patients at surgical resection of a primary lung tumor. And what we did was we carried out broad a quantification of the immune compartment of this normal tissue. And we looked at a range of different immune cell types. And surprisingly, what we found was actually the phenotype of macrophages within this normal lung tissue of people is associated with the levels of PM2.5 at the postcode level of which these patients live, possibly suggesting that that macrophages are a key responder both in the mouse and in humans. And then, so to further dig into the mechanism by which this is happening, we carried out RNA sequencing of the epithelium following exposure to the particulate matter. And what we found was that there was evidence that the AT2 cells were adopting a progenitor cell state. So we saw a possible keratin 8 expansion within the AT2 subpopulation. We saw macrophage recruitment signals, and we saw some epithelial alarmins. And so then what we wanted to do was compare what this might be happening to in humans. And so we started this fantastic collaboration with Christopher Carlston in Vancouver, who's got this IRB approved study, whereby a clinical crossover study whereby healthy never smoking volunteers are exposed to either particulate matter or 2 weeks later filtered air control. And then bronchial brushings and RNA-seq was carried out on these samples. And so we've got paired samples to understand what the acute effects of particulate matter are on the lung transcriptome. And this was a small and noisy data set, but what it allowed us to do was we could directly compare the fold change within the mouse lung transcriptome response and the human lung transcriptome response. One of the things that really jumped out at us was IL-1 beta, which has been known to induce and is sufficient to induce this altered alveolar type 2 state. And recently, or several years ago now, it was found that in this hypothesis generating study that there was patients at risk of cardiovascular disease were given canakinumab, an anti-IL-1 beta blocking antibody. And the incidental finding was that there was a dose-dependent decrease in the number of lung cancer incidents, suggesting that anti-IL-1 beta might be mediating this process. And so we carried out the human experiment in the mice, and we blocked anti-IL-1 beta during the exposure to particulate matter. And what we found was this was sufficient to significantly decrease the tumor formation induced by particulate matter. And then finally, onto the kind of third prong, is that if these tumor promoters are acting in a non-mutagenic fashion, then it suggests that these pre-existing mutations must be present in latent cells within lungs over time. And so we explored whether these EGFR mutations are present in normal tissue. And to do this, we used 3 clinical cohorts. We used the TRACERx study. So these are patients who have lung cancer, and at surgical resection, an adjacent normal was taken. We used the PEACE autopsy study, whereby these patients never developed lung cancer in their lifetime. And finally, we worked with James Gregory in Colorado to study a cohort of patients who get a lung brushings of a contralateral lung for people who are undergoing screening of a suspicious lung mass. And what we found was actually that these EGFR mutations are surprisingly common, that in all 3 cohorts, we found that pretty much 18% of samples contained EGFR mutations. These were present at very low variant allele frequency. They're very rare and have only really been able to detect due to improved technologies in sequencing. And we just focused on oncogenic driver mutations, and using Bayesian inference, we calculated that about 1 in 600,000 cells within the lung contain an oncogenic EGFR mutation. And one of the things that this larger cohort of normal tissue allowed us to do was explore clinical associations between the presence of EGFR mutation. And so what we did was, within lung tissue that's been well documented, that you can get anthracosis, this black pigmentation, which is associated with environmental pollution. And we saw no association with anthracosis and smoking status. And so we explored firstly whether anthracosis, just the presence or absence of anthracosis, was associated with having an oncogenic EGFR mutation. And we saw no association, suggesting that anthracosis is not inducing these mutations perhaps. But what we did find was that the variant allele frequency of these ultra rare clones was significantly elevated in samples with anthracosis. And how we interpret this is that in samples with anthracosis, perhaps those cells are beginning to expand and proliferate. And so just in summary, what we think is that over time, these pre-existing oncogenic mutations arise within histologically normal lung tissue, such as the EGFR mutation. And then what we think is that particulate matter via IL-1 beta can induce progenitor-like cell states within these primed cells to drive some of the earlier stages of tumorigenesis. And I think this is quite exciting, that it offers opportunities to really understand the molecular mechanism by which environmental factors work. And it allows us, I think shows quite nicely that we've tried to incorporate these multiple different models and using the mouse really to its strengths, where it offers a controlled system for when we induce the oncogene, when we treat it with particulate matter or other factors. It allows us to robustly study early tumorigenesis in an immune proficient model, but it has a number of weaknesses. It's got a very small disease burden. The maximum tumor size is a few millimeters. It's a very short time frame. It poorly recapitulating the decades of exposures and variety of exposures. And then also the obvious differences between species and translation between this. But I think it can be really useful to help define the questions that we can then go back and address using our clinical cohorts. And this took it was a huge team effort with international collaborations throughout the world, which I'd like to acknowledge, and of course the funders without which this wouldn't be possible, and the patients and their families. So with that, I'd like to stop and happy to answer any questions.
That was great. Nice job. We have some questions that are coming in online right now, so I'll try and paraphrase them. And probably this will be, a common question in the theme here of the session, is that two people are asking roughly around the range of the fact that genetic background or humans vary quite a lot. So how well do you feel the responses that you're observing in your mouse models compare to what you're seeing in human studies? And obviously there's some correlation you were seeing with the UBC study, but it'd be interesting what your general impressions of that are.
Yeah, so I think there are clear differences between what's happening in the mouse and what's happening in the human, but the mouse exposures have really allowed us to refine the questions that we can ask within the clinical cohorts, and yeah, the kind of complexity that occurs within a human, and really use a reductionist system to ask simple questions.
So I guess another question in a related vein is that you looked at 3 different mouse models with different genetic backgrounds, and 2 of them being EGFR related. Do you think there's a benefit to exploring additional genetic backgrounds, or do you think actually just expanding the sample size with more isogenic lines would be useful?
Yeah, I think these are very I guess simple genetic models, in that you're inducing a single oncogene change and studying what happens between that and the pollution exposure. So it's starting to perhaps explore the effects of urethane, or these kind of chemical carcinogenesis models where you're going to induce a range of different mutations. So I think it'd be quite interesting to explore in these more broad chemical carcinogenesis models. Do you get the same genetic profile that you would, or the same response that you would in the control and the treated samples, or is it selecting for different genetic or other phenotypes?
And I guess in that vein, the PM 2.5 is a pretty gross description of an environmental exposure as well. So there could be lots of underlying chemicals or other types of things that are present there, which might aggravate this more than others. Is that?
Yeah, I think this is really a kind of initial study to develop the model. And now we've developed the model, now we can begin to explore, is it the size of the particles that are important? Is it the chemical composition of those particles, and explore which one might be driving the effect?
There's another question online here. It's asking about the EGFR driver mutations. So were they point mutations, indel, structural variations? Are the mutations by any chance C to T transition mutations?
Yeah, that's a good question. So within the digital droplet PCR, we looked at the 5 most common oncogenic drivers that occur within people, which does include the exon 19 deletion and the L858R mutation. Off the top of my head, I feel like this is something we have looked at. I can't remember whether they fall within the C to T, but I feel like they don't.
Okay, yeah. Another question online was, someone was curious to know what you found about KRAS in normal lung tissues.
Yeah, so yeah, I didn't really have time to go into it, but we actually found that KRAS mutations were present in 50% of the samples that we studied. It was a smaller sample set rather than the several hundred that we looked at for EGFR, but I think within around 50 patients, around 50% of them had KRAS mutations, which I think raises some really interesting questions about disease progression.
Yeah, I was also really interested in that, and just what kinds of other background somatic mutations that you would find. So do you think like the 18% rate that you find for ERAF is just representative of what you'd find for any other type of mutation, or is it elevated?
Yeah, yeah, that's a really interesting question. I think the I think there's some evidence, for example, Notch1 in the esophagus, that the selection pressures within normal tissues can be quite different to cancer. For example, Notch1 is selected for within normal tissue. And I think within the intestine as well, with NFKBs, mutations that are selected for may be slightly better at dealing with inflammation rather than forming a tumor. And they're not always necessarily completely overlapping. And I think, again, this comes down to technological difficulties in studying normal tissue, in that it's very difficult to go very deep and very wide. So we've been using a panel-based approach, which means you're always looking into the lamplight, but we're hoping to go broader and look at more genes and see which is 18% the normal for everything, like you say, or perhaps is it what's higher and what's normal?
Yeah. Well, even I would imagine like increased granularity for the transcriptional profiles might be interesting to look at too, because it can be hard to understand if the IL-1B, and excuse me if I got the gene name wrong there, if that signature is actually macrophage related, or is actually something intrinsic to, a specific cell type underneath it.
Yes. So one of the experiments which we've done recently, which was really quite elegant, was we took macrophages from mice which have been exposed to pollution, and then did a co-culture system with EGFR mutant AT2 cells. And what we found was that was sufficient to induce both functionally and transcriptionally the kind of altered AT2 cell state. And so what we think is that the key mediator of the signal does appear to be macrophages, at least within that readout. But yeah, the going from the complex immune of the lung to different cell types, and which cell types are most important and when, is a really good question.
Yeah. A couple more questions here online. So someone was saying, if I was looking correctly, in the mouse experiment, the mutation levels were higher with exposure, even though not statistically significant. With such a small number of mice, can you say with much certainty there is no mutational effect of PM 2.5?
Yeah. I mean, we carried out whole genome sequencing of these tumors, so we were limited by costs. I think it did appear that there's possibly a trend, but I think the rescue also quite clearly demonstrates that the tumor promoting effect of particulate matter that we're seeing doesn't seem to be arising due to the possible mutations. And then when you look at the signatures, it's exactly the same signatures, suggesting that perhaps particulate matter is not acting in this way.
Yeah, and I guess related to that was, if you have any hypotheses around why UV-related cancer mechanisms versus air pollution cancer mechanisms might differ, like why they might actually, have different mutational spectrum, or different genes that are involved.
Well, I think these effects might be quite tissue specific, and so I guess that's probably the obvious, that how the skin responds to the challenge of UV light is going to be markedly different to how the lung responds to particulate matter irritant. So I think that is
the type of DNA damage that probably occurs from those, how that's initiated.
Yeah. And I think it's perhaps it's whether something may be inducing low-level mutations. I think there's still further work to be done, but I think the important thing is that this offers a potential additional mechanism by which these factors are acting, which offers possible therapeutic intervention and could be stopped.
So I want to ask one last question. I think it's the best question to end with here, is how do you think this research translates to other cancer types, like breast cancer, as an example?
Yeah, a really good question. I think this is, as I said, I think this is an initial study opening up a really wide range of, perhaps, as I said, we saw KRAS mutations in 50% of these lung samples. So I think this goes broader perhaps than just particulate matter, and could offer a potential mechanism by which multiple tumor types, multiple factors might be acting. And so I think it's really important. We've got a whole range of mutagenic models to understand is a carcinogen acting as a mutagen, but we're really lacking tumor promoter assays. Is come to quickly test a range of models and environments as to whether something could be acting as a tumor promoter.
That's great. Well, thank you again. That was an excellent presentation, and for answering all the questions that came from attendees as well.
Thank you.
So at this point, we'll move on to the next talk, and we have Matthew Duvall from the University of Virginia.
Thank you. If I just share my screen. So I'm a senior scientist at University of Virginia, and I've been working in the colon organoid space now for a period of 5 years, where we look to model environmental factors that increase the risk of colorectal cancer. So the reason for the colon organoids, or should I say what our interest in the colon organoids, is their direct relationship to patients with regards to the cellular pathways and things that come out of it. Sorry, that was a terrible explanation. Colon organoids are derived from individuals at the time of routine colonoscopy. They are a great representation of the colon crypt. We take colon organoids from healthy individuals in our lab. We're really interested in the colon epithelium. We take crypts from these organoids and we throw them into wells containing various different media conditions that were defined in a publication in 2011 by Toshiro Sato and Hans Clevers at the Hubrecht Institute in Utrecht. The reason why these organoids are fantastic is because they contain a lot of the cell markers that are present within the colon crypt, such as CHGA, which is a primary cell marker for enteroendocrine cells. Some of these markers you can't adequately or easily get from some existing cancer cell lines. But most importantly for us, one of the major drivers is that these kind of mini guts have been suggested to be able to be grown indefinitely. Once you have them and you've established them, you can use them for potentially a lifetime, which has motivated the research in our lab for the establishment of a UVA biorepository. This was work that was originally done by Dr. Casey, and following his retirement, we transitioned this organoid resource to Dr. Lee, and I've been helping him try to establish this and improve upon the work of Dr. Casey as well. From Dr. Casey, we developed 140 genetically independent colon organoid lines, both left and right colon organoids from these individuals. And our work now is really to try and generate this, really ramp it up and increase the number of samples to about 500 lines, so about 1,000 organoids total. And this is primarily because of some of the findings that we've identified previously from our analysis in colon biopsies, where we see, if you look at the global transcription profile of colon biopsies and plot them on a PCA, you can see that colon biopsies from the right and the left colon in red and black respectively are quite dissimilar. Whereas colon biopsies from the transverse, which separates the two, actually fall relatively in the middle for females and for males as well. So this is really quite interesting, and it falls in line with this increasing understanding that we have that left and right colon cancer is very different. To assess environmental risk factor exposures, I've developed a framework over the last 5 years for the colon organoid analysis. And this framework has been performed on the average risk population. So we take colon organoids from individuals at the time of routine colonoscopy who don't have a family history, who don't present with a high degree of polyps, and a number of other exclusionary factors. We expose them to various different colorectal cancer risk factors that have been defined in the epidemiological literature for a period of about 24 to 72 hours. We perform bulk RNA sequencing on these, and because of the size of these studies, ends of 34 to 40 lines, we're able to do some more complicated analyses By this, we've been doing machine learning approaches to estimate the cellular environment and differences in cell composition that occur following exposure to various different carcinogens or chemopreventive agents or potential chemopreventive agents. We're able to identify changes in populations, specific populations, and we're able to incorporate these self-scores as covariates into our differential expression analysis and into our network analysis as well. And one of the reasons we've been doing that is because we find that by addressing this kind of extensive cellular variation, we can actually better replicate some of the findings that we've observed to be kind of gold standard from previous other studies. So the primary focus of our research has been to identify the molecular mechanisms that drive the effects of observational epidemiological studies. We do really large-scale studies with this when you consider that the typical organoid study is usually about 3 to 5 lines. And we're really just interested in the organoids generated from healthy colon biopsies. We're looking at chemoprevention and colon cancer risk. It's been really successful over the last couple of years. We've published 4 different studies so far in these kind of large-scale exposures on aspirin, calcium, ethanol, and carcinogens. And very briefly, I'm just going to talk to you about kind of some of the consistent themes that we've identified before then going more detail of the carcinogen exposure. One of the really exciting things that we've identified in these studies is that organoids derived from the right and left colon respond very differently to drug treatments. So we've taken organoids, these large-scale studies, and we've exposed them to alcohol, for example, and to aspirin, and we found that organoids from the left colon actually exert have a much larger number of differentially expressed genes associated with that response than organoids from the right colon, while the inverse is actually true for aspirin treatment. And this is quite exciting because there's been a number of different studies where we found a greater chemopreventive effect for aspirin in right than left colon. A number of studies have looked for high to moderate alcohol consumption in the left colon and found that it has a greater effect as a risk. So we're not just seeing that we can identify interesting mechanism from these studies, these colon organoid studies, but also that they translate in terms of that kind of high level, which side of the colon is really being affected. As a kind of one thing that will come up a couple of times in this will be that all of the exposures actually led to strong changes in epithelial cell composition, with risk factors driving more proliferative states, these increases in these transit amplifying zones, and some of the chemopreventive agents leading to the opposite effect, this reduction. We found a pretty reasonable commonality between the number of genes that were found to be differentially expressed in these environmental exposures and those that map to colorectal cancer through GWAS studies. And that can be seen here, whereby the differences in, we don't just see a large number of differences in gene expression, but we also see that some of the genes that pop up across different exposures are actually quite similar. Really kind of suggesting that there's a common theme for these drivers in cancer risk. So with that in mind, I'm going to talk to you now a little bit more about one specific study in carcinogen treatment that we published in late 2021. We exposed healthy colon organoids to a custom carcinogen cocktail for a period of 24 hours. Following that, we performed bulk RNA sequencing analysis. These carcinogens were found commonly in cigarette smoke, and that's what we were really interested in trying to model. They were found within the colon following individuals who smoke cigarettes, should I say. And we found pretty extensive number of differentially expressed genes following this exposure, as one might expect when you expose healthy colon to a range of carcinogens. We identified 738 differentially expressed genes with some pretty obvious targets coming up as being overexpressed following our analysis. But one thing that we had issues of is, okay, we find almost, so many differentially expressed genes, but how do we prioritize these? How do we really garner some mechanism from this? One way we could look at this is just to go through standard pathway-based approaches. but they can often be quite biased by what significance level you decide to attribute to these kind of pathways. If you decide to use a cutoff at FDR 0.1 or 0.01, you might find different pathway enrichments, et cetera, which can really kind of bias your results. So one of the things that we looked for was trying to identify potential interplay between gene and environment, and prioritize plausible targets as ones that were popping up through colorectal cancer GWAS loci. We found that 61 of the 739 genes were within a 1 megabase region of colorectal cancer GWAS. This actually represented an enrichment for CRC GWAS genes in our data set. And you can see here just the 5 most differentially expressed CRC GWAS genes in our analysis. However, while that's really exciting, genes really rarely act in isolation. If you knock down APC, it's going to lead to a lot of other gene expression differences that are very important to cancer risk, and it's not just APC that's driving this change. Whereas single gene analysis can fail in helping us understand that co-expression, it's also attributed with a pretty high false discovery rate burden. And so what we were really interested in is defining networks based on gene-gene correlation structures, so WGCNA. This has been mentioned a couple of times now, but WGCNA is a concept whereby you group genes based upon their correlation into modules that are really highly co-expressed. And following the establishment of this network, you then take these individual modules and their module eigengene, their average expression, and correlate them or associate them to your trait of interest. So one of the reasons why this is really helpful is because we're not just looking at gene-gene correlations that are limited by some arbitrary p-value that we select to consider significance. This is one of the examples of our networks from the carcinogen treatment, just to give you an idea of how these structures look like. But primarily, one of the important factors that I want to try and consider for these analyses is that the most significant gene is not necessarily the most important. For example, a small change in a transcription factor like MYC might have really wide-scale differences in this gene co-expression that could have some major biological implications, but it doesn't have to be hugely and dramatically changed to exert that effect. So our network analysis, it revealed 7 modules that were significantly associated with carcinogen treatment. And the kind of module eigengenes, the average expression here, the differences in average expression can be seen here. I'm going to bring up one particular module that we were really interested in, the coral module. The average expression of the genes within this coral module were reduced following carcinogen treatment. And what's exciting about this module particularly is that it's enriched for pathways related to mismatch repair. And in fact, several of the genes that were present within this module were not identified in our single gene analysis, so were only nominally significant. They were really highly relevant to the module, both in terms of mismatch repair, and they were not actually amongst the top 10 hub genes that I selected here for the publication, but they were all within the top 20. Now, for those of you that don't know, MSH2, MSH6, and MLH1 are key driver genes for Lynch syndrome, which is a genetic predisposition for colorectal cancer. People who develop Lynch syndrome related cancers often have tumors from the microsatellite unstable high pathway. MLH1 is also a driver of somatic development of this MSI high tumor pathway. And these smoking carcinogens we were able to identify led to these major differences, this downregulation of these mismatch repair genes that is well in line with what we kind of were understanding with smoking, that smoking is more strongly associated with MSI high tumor development. And importantly for us, we were not able to identify these differences both at pathway level or in single gene level prior to this analysis of WGCNA. So how we then can consider further integration of the environment. One of the things that we've been looking to do is replicate our findings in real world. We developed a Barsuva-seq data set in collaboration with Barcelona with Ricky Peters at Fred Hutch and Jim Gauderman and his team at USC. And this data set consists of about 470 normal colon samples with pretty extensively matched epidemiological data. So we were able to identify that the carcinogen-related findings that we had from our colon organoid system were pretty largely overlapping what we were seeing in individuals with smoking history, which was great. I think it's really important to try and replicate these findings in real-world cohorts. We also saw that on the kind of macro level of this, carcinogen treatment led to an increase in goblet cells. And we note that this was also something that we were able to see in Barsuva-Seq, which we thought was really exciting as well. So finally, I'm going to go on with some things that we've been doing since then, a secondary follow-up of this data set, which we're hoping to publish pretty soon. And that's been motivated around some of these publications that have come out on cancer organoids, where they've shown that intra-individual levels of variation might be useful in terms of predicting personalized response to chemotherapeutic treatments. Now, all of our research is done in healthy colon, but what we were really interested in doing is determining whether some of our lifestyle variation covariates that we have collected for these individuals, whether they influence the extent of the modular eigengene expression and association with our trait of interest, so carcinogen treatment. And this can be seen kind of broadly here, whereby for each individual, you're able to capture a average expression profile of all the modules within that gene, this modular eigengene, this first component of the module. And then you're able to relate the extent of that expression to your trait of interest, which is where you get your significance. However, if you're able to catch such intra-individual levels of variability, you can also start to look to see how they may be affected by factors other than your trait of interest, your carcinogen treatment. And that's exactly what we've done recently, where we've looked to see if individuals who had a previous smoking history were differentially primed to respond slightly differently in these in these very specific ways, whether these robust modules were actually also influenced by an individual's pre-existing history. And then actually one of the final sections going back to colon location is that during our single gene analysis, unlike ethanol, calcium, and aspirin, we were not really able to find any major differences in left-right sidedness in terms of the magnitude of differential expression response. However, when we've kind of followed this up with our network-based approaches, we were able to determine that there were significant differences in location for this modular eigengene expression. And just one example of this can be seen for the coral module, this module that was related to defective mismatch repair genes that drive microsatellite instability. We saw that this was more greatly reduced in right than left colon, that all samples had a reduction in, all samples led to a reduction in genes within this module. However, this reduction was more significant in right than left colon organoids. What's really exciting about that is that MSI high tumor development is primarily seen in the right colon. So we're really excited about the implications of these further developing these kind of statistical frameworks. To do that, we really need to assess the interplay between genetics and environment in colon epiphelia, and also to further integrate extensive phenotypic data. We should also be considering, one of the major limitations of our studies have been that they've been primarily in European American sample sets, and we've had some issues in trying to recruit African Americans into these studies. But we're now really pushing to increase cohort power and diversity, and hope that this will allow us to really explore new avenues of research. There are some limitations with these kinds of studies. I think you have to choose your complexity of interest. We're really interested in colon epithelium. However, I think it'd be very challenging to ramp up the number of samples if we were also interested in something like co-culturing methods for immune cells or fibroblast cells, and to look at that kind of cell-cell communication. I think that's, at this stage, just not something our lab certainly is going to be able to do. When you're considering increasing the number of lines, you also have to think that each organoid line has to just be in culture for a while before it can be exposed to anything. And so just managing that kind of workload puts limitations on how long you can actually expose these cells to different environmental factors. And you have to make sure that you're controlling for variation in phenotypic variability for within these samples so that you're not biasing your results downstream. But with that in mind, we've now generated extensive food frequency questionnaires and have access to lifestyle factors, medical records, and health in our colon organoids that are being established since joining Dr. Lee. And this has been possible because we have a really great clinical team that collaborate with us that are really willing to collect biopsies. It takes a lot of time from them. It also takes a lot of time from the nursing staff who are always busy, who are collecting blood, stools, toenails, urine, etc. And so that, really grateful for these people. We need staff that have very good at interacting with patients, who are good at following up as and when they say, and who can provide clear information for consent. And we have to rely on environmental data and the fact that colonoscopies are subject to variability around the individuals. Sometimes they get cancelled, etc. But with all that in mind, I'm very excited for the future of colon organoid research and for the future of environmental risk in general. I'd like to really thank all of our collaborators as well, and particularly the gastroenterology team, Dr. Powell and Dr. Yoshida.
That's great, thank you. So we'll open up to questions now. People can submit questions through the Q&A. We'll just start with a few that have appeared already. One person's actually looking for a little bit more understanding of how you make organoids themselves. And so I was wondering if you could take a few seconds to describe the experimental process there.
Sure. Colon organoids can be established now actually from frozen biopsy, even as long as you do some pretty early pre-processing steps. You can, a big part of the colon organoid establishment is making sure you have a clean prep. Some of these, because they come from colon tissue, if you have some major problems with that kind of prep, you won't be establishing them. There'll just be contamination and they die. But the process for growing colon organoids has been around for about 10 years. It hasn't really changed in that time. A major notable change was a Stappenbeck publication, which I can provide in 2011, which followed on from the Toshiro Sato paper. But I think the main thing really is getting them being ready at the clinic at the time in which colonoscopy has been taken, rushing them back on ice, and cleaning up the biopsy before you just start to shake off the crypts in different media.
Yeah, and how long does it actually take to culture them? Because I know there's a lot of variability with different organoid models.
So I think for us, you can, because we primarily want to get as many colon organoids, as many of the same sample at a low passage number as possible. So from the time of colonoscopy to the time that we are freezing them down, it takes probably about a month per line.
Let me see. Oh, that's pretty good. And are there issues with storing the biopsy material and generating lines later, or is that generally not a problem?
We've had, as long as you do the first few like PBS washes from on tissue prior to freezing, and you are making sure that it's a slow freeze, you can generate organoids. I think the slow freeze really is also very important. Slow freeze in -80, just to make sure that you're not damaging these colon organoids, these crypts prior to colon organoid establishment.
I see. Well, one person's asking a related question about whether or not there are things like age or smoking or other environmental types of effects which might impact your ability to develop an organoid line.
So that's a really interesting question. I would say that our organoid establishment protocol now is, it's been something we've been doing for about 5 years. The biggest problem that we've had really is the cleanliness of any kind of prep. In general, so that sometimes means that kind of rectal biopsies or left colon have slightly, can be slightly poorer, and that's usually where we see some of the issues, but we haven't looked to see if environmental factors have caused any major issues.
I think related, and you touched on this a bit, that in terms of the data that you're collecting, there are still challenges with expanding it across different ages, different genetic ancestries. What kinds of challenges are you facing there? What do you see are opportunities to enhance representation of these types of lines over time?
So I think one of the issues that we have here is for age, certainly, is that routine colonoscopies are performed from the age of 45 onwards. And so you do have that kind of limitation in terms of there's usually a reason why somebody's coming for a colonoscopy earlier than that age. And so you have to kind of then check to see, well, how relevant are these to the average risk population, which we're interested in. So there is certainly like an early cutoff that we have for colonoscopies. And at the same time, there's usually a late cutoff. People past the age of 90 are not very frequently getting colonoscopies. So those do, limit the range and scope of what we can do. With regards to incorporating African-American populations or other ethnic minorities, one of the big things that we've had issues with previously is we've had issues in just, this is part of a larger study that we've developed for biopsies where we're collecting 7 or 8 different tissues from these individuals. And not every tissue can be collected with organoids, just because we just don't have the manpower to do that kind of thing. However, now what we're trying to do is to ensure that we've had a systematic look to see where are the missing parts, where are we really missing representation in our data set, whether it be African American, whether it be different age groups and categories, for example. And they're really the priority samples that we take from the colonoscopies now as well, to try to fill in those gaps.
So I'm going to generalize another question here. So someone's asking about how you translate exposures in natural human populations into that organoid model in terms of concentration. But I think there's a lot of variables there, not just concentration, but then how you do it over time, how you try to get to that physiological exposure, and maybe you can comment on that a bit.
Sure. I think so. This is where interdisciplinary collaboration comes in as well. We can only model kind of, for example, for concentration, we can only model what we understand to be a relevant concentration in the colon based upon what's currently available. For some studies, we had a collaboration with David Drew, who's a real leader in aspirin research at Mass Gen, and they have data on aspirin concentrations in the colon, for example, following aspirin treatment. So that kind of thing is very helpful. You can be pretty confident in how you're modeling it. I think that that's a primary limitation of any study. And I think that that's where, extensive collaborations would be very beneficial to make sure that we're modeling things that are like really capturing what's going on specifically in the colon.
And another question that I had was actually, I've seen in the literature people taking organoids and creating xenograft models, particularly around these patient-derived organoids. And I'm wondering what you see are the relative merits or disadvantages to those types of approaches. Is that something you're pursuing?
It's not something we pursue. It is something I think is obviously very relevant. I think it's great to one of this comes in with kind of translation as well, but also one of the final points that I mentioned with regards to cell-cell communications, we can only really look at very specific number of cell populations in a model system that exists within a dish and not within a living organism. So importing these patient-derived organoids into various different mouse models, I think is very beneficial to really help to drive our understanding in disease mechanism.
And then maybe just one last question here. So I noticed that you had enrichment, but it was pretty modest enrichment for your colorectal cancer GWAS into the genes that you're studying. Are there, do you see particular challenges that are there with intersecting GWAS data into these types of model systems, or are there needs for tools or additional data that could help pinpoint the relationship between that genetic risk, but then also the observations you're seeing in terms of exposures in your system?
Yeah, so for this study, actually, the enrichment was fairly modest, as you say. It really just got into that kind of significance cutoff. I think one of the things that as we start to ramp up these study sizes, we can be able to stratify out left and right differences. There's been a couple of studies, for example, by Ricky Peters at Fred Hutch, where they're seeing different GWAS findings for different regions of the colon, and we're seeing different genes and different loci that are popping up based upon colon location. And so I think, that kind of fine tuning what we understand as genetic risk is really important, and also important for the colon organoid system as well.
Yeah, that's excellent. Well, thank you again for your great talk and answering all these questions. And I think now we're moving on to the panel session.
Thank you.
So I'm just going to take the opportunity to introduce our 4 panelists. Each of them then have 5 minutes to give a general overview of their work based on what our session topic's about. So I'd like to introduce Rebecca Fry from UNC Chapel Hill, David Reif from the NIHS, Francesca Luca from Wayne State University, and Justin Colacino from University of Michigan, the School of Public Health. And Rebecca Fry will be going first for the intro.
Great, thank you so much. And thank you to the organizers for inviting me to participate in the workshop. In my time allotted, I'd like to share with you how we're using multi-omic data sets to inform health and disease characterization as it relates to environmental chemicals. And I believe that many of the strategies are highly relevant to multi-omic use in cancer biology. In our lab, we focus on chemical exposures, particularly those that occur during pregnancy, or even more recently, some of our studies are pointing to chemical exposure that happens prior to conception. So either these in utero or preconception exposures as they relate to pregnancy complications, fetal development, and later child health. The chemicals we focus on span from toxic metals to organic chemicals such as per and polyfluoroalkyl substances. All of the chemicals that we're working with in the lab are of significant public health relevance. They're known to impact the health of pregnant women, the developing fetus, and children both in the US and also globally. And with relevance to this workshop, many of the chemicals that we study are also established carcinogens. So we know from numerous studies that exposure to chemicals or other stressors during the prenatal period is associated with adverse health effects assessed early in life, as well as later in life. This concept is, of course, known as the developmental origins of health and disease hypothesis, or DOHAD, and that there could be long-lasting health effects from early life exposure does suggest potential for cellular epigenetic reprogramming resulting from exposure to these chemicals. My lab focuses on the placenta as a target tissue of interest for studying chemical disease relationships. And our interest in the focus on the placenta is, of course, because of the essential role that the placenta plays in fetal growth and development. It's key to separating biological components of maternal and fetal systems, such as separating blood supplies between the mother and fetus. But the placenta also creates an essential interface between maternal and fetal systems. So providing critical hormones that are transferred to the fetus that are essential for proper fetal development. In relation to the environment, the placenta can also serve as a transporter and transducer of toxic substances, where maternal exposures are often directly related to fetal exposures. As a result of this and the fact that dysregulation of the placenta is associated with early and later life disease, we broadly call this study the placental origins of health and disease, or POHED. In relation to the workshop today and model systems of study, we examine placentas that are derived clinically from human populations, primarily located in the United States. And we also use classic toxicology-based approaches using placental cells as our model to study in vitro the effects of chemical exposures where we can control concentrations as well as time. In terms of omics data that we're analyzing, the general approach is to use RNA sequencing to gain an understanding of disrupted biological pathways in relation to chemicals in the environment. In addition to these, to complement the RNA sequencing, some of our recent studies also include placental proteomic data to get a sense of the translation of the messenger RNAs. We then ask if these biological pathways are under epigenetic control, focusing on CpG methylation levels as well as microRNA expression. And some of the work we've carried out using multi-omic strategies in the placenta highlights that there are biological pathways that are disrupted by chemicals in the environment, such as arsenic or cadmium or PFAS. To provide some examples, we've shown that arsenic is associated with dose-dependent changes in gene expression, as well as dose-dependent changes in DNA methylation that controls the messenger RNA. And often these data point to common biological pathways, such as those involved in dealing with cellular inflammatory responses. We've also shown that these multi-omic data in the placenta can be used to predict health outcomes later in life in children, such as autism spectrum disorder. More specifically, we've identified genes where mRNA expression, the CpG methylation levels for those target genes, and the targeting microRNAs all predict autism later in life. To me, these data indicate the placenta represents a recorder of the in utero period, which we know is strongly associated with later life health. So bringing this back to the topic of the workshop, in terms of ways that multi-omic data could be used in the context of cancer, I think about the following. First, that multi-omic data could be used as biomarkers of environmental exposure. So for example, are there specific epigenetic marks within tumor tissue that could be used to indicate past exposure to chemicals in the environment? Second, could multi-omic data be used as biomarkers of health outcomes? As an example, could we use epigenetic marks to predict risk for cancer or an individual's responsivity to chemotherapeutics? And finally, multi-omic data could be used to inform biological mechanisms of disease, so specifically to identify which biological pathways are altered in tumors. And these data would likely expand the potential opportunities for clinical treatment for cancer. So with that, I've probably used up my time. Thank you again.
Great, thank you. Yeah, our next panelist is David Reif from the NIHS.
Hi, yeah, thank you. I'm glad, Rebecca opened with talking about the multi-omic approaches and also, developmental periods and all those kinds of important considerations, because I'm going to leave that out a little bit because I know she was covering it. So I recently joined NIH as chief of the predictive toxicology branch, and that sits within what is newly renamed as the Division of Translational Toxicology at NIHS. And there's some reasons for that renaming, but we used to be the division of the National Toxicology Program. People I think are familiar with NTP. We still support that, but I think as a translational toxicology division, we have a decidedly translational bent to our research. So I'm going to comment on some things of how I actually view translational as also being perhaps transitional. So going between layers, multi-omics to me doesn't necessarily mean, and I think we've seen that in lots of presentations, that you have to get the same information, same type of information at all levels. But it's how can you use information to start building these bigger models, right? And I think, I have copious notes from the last couple of days of fantastic things I need to read and study up on more. But I'm excited about those. So I'm going to, for this session, I'm going to tell you some models and functional approaches to address G/E that we're using from exciting work, I think, here at NIEHS, as well as continuing research with academic partners from my time as a professor at NC State. So, the intro, I think, by Dr. Montgomery included mention of zebrafish and translation directly to the clinic, but also through, mammalian models and organoid work, as the other speakers pointed out. And so I think it's really important to consider the number of steps and the number of phases between each translational step, right? It doesn't necessarily need to go directly from one to the other, as I think we've seen. But just to restate and emphasize that, I think that, in our branch, we also have folks working on NICEDM, which is focused on new approach methodologies, or NAMs, right, to change the way that traditional tox testing is done. So we have studies like that through traditional in vivo studies, and also even geospatial work with a spatiotemporal exposures and toxicology group. So you're bringing all that information together, I think emphasizes how, or my experience in trying to unify these data, is that already you see that data are generated at wildly different rates, and at those levels, different resolutions, spatial, temporal, all those things. And certainly the population variability is a big one, right? And so I think there was a workshop a few months ago on NAMs to address population variability. And that one had a lot to do with organoids and human donor models and things like that, bringing that kind of thing into the space. I think there's a lot of progress there, and I'm excited for that. But I also think that the information generated at each level, you can use it as a basis for other predictive applications. So, generative adversarial networks, or GANs, all kinds of machine learning approaches. They're blurring the lines between what is strictly where a study falls in the continuum, right? Because I'm using data from one level to predict and insert, and it's not imputation, it's a little bit different things at other levels. So I think there's a lot of excitement there. I'm particularly interested in models for behavioral gene environment interactions, or G/E, because I think behavior is a quantitative trait that all the geneticists in the room will be very excited about how wonderfully the statistical tools are available for QTL mapping. And this is something that's, since the beginning of statistical genetics, has been something that people have been working on. So I think some of these models that are amenable to high throughput things, I'm thinking in particular of zebrafish studies, and some recent work I've been doing with Robin Tangway at Oregon State University and Fred Wright at NC State University, using this high throughput model to actually test for population susceptibility differences, because they are, if you have an outbred population, they're genetically distinct from each other. And so it's much like a human population. They're not aligned. We don't know the genetics, but they are diverse. And so, we started by asking questions of what in the exposome was actually showing signals of differential expression, instead of just saying my favorite chemical and I want to chase it down, right? That's a different approach. So you can use these kind of throughput things to balance these focus studies. And I think behavior is a really good endpoint from a public health perspective, because to put on, the most regulatory toxicology kind of translational hat, the point of departure is important. And I think we often see that behavior is an early indicator of things going on later. And I'll point you to some recent work from our group where we found a behavioral signal of population susceptibility differences at a low or medium dose. And then as you increase the dose, that signal vanished, right? The signal doesn't go away. It's just that the compound then becomes a poison, right? So we all sort of know this, but you can use these systems to start detecting those things that are signals of G/E to kind of follow up on that. And the last point is, with these things, I hope that the functional exposures approaches can be amenable to realistic exposures. And I'm excited about things from in vitro to the co-culture models in that sense, because we can do mixtures, we can do realistic things, we can do temperature times a chemical agent, right? That's all really exciting. So I'm glad to see that talked about. And thank you again to the organizers of the workshop. Again, I've learned a great deal already, and I have lots to study.
That's great. Thank you. Next up is Francesca Luca from Wayne State University.
Hello, and I would like to start by thanking the organizers for the opportunity to participate. It's been 2 days of really interesting and thought-provoking scientific discussion, so thank you. So I'll give a brief introduction of my research interest and some of our experience that is of interest for this group and this discussion. So we've been studying genotype by environment interactions, both in molecular and complex traits in humans. And we use different types of omics, both with in vitro and ex vivo samples. So we've been considering a variety of different exposures, from chemical exposures to psychosocial factors and microbiome exposures. And we measure our molecular phenotypes in a variety of different cell types, from blood samples to primary cell cultures, as well as colon organoids. So there's really a lot of overlap with some of the things that we've been working on and thinking about in my group, and the discussion we've been having in these 2 days. But what I would like to focus on is what has emerged both in these 2 days and from Stephen's introduction is one of the major challenges when we want to focus on genotype environment interaction studies. And so that's challenges that these studies are multidimensional. So at the same time, ideally, we would like to be able to interrogate a large number of exposures. We would like to be able to sample extensively genetic variation. And ideally, we would like to use multi-omics approaches. So when we consider this complexity of studies and how to design them in our group, we have developed approaches to study allele-specific expression and allele-specific effects in also other types of omics data. So the major advantage of these approaches is that we don't need to analyze large population samples. So we can essentially reduce one of those dimensions, the one that comes from the population samples. And this allows us to focus on a larger panel of exposures than otherwise we would be able to do. And so with allele-specific approaches, we can essentially focus on a small sample size in the order of even less than 10 individuals. And we can either use naturally occurring variation in this case, or we can use more recent approaches of synthetic biology, which are this massively parallel reporter assay, where we can essentially design regions of the genome with genetic variation and then test them in specific cell types. And I'm going to go a little bit more into the details, or not really the details, but kind of give a little bit of insight into how these two experimental approaches work. So the underlying concept behind allele-specific approaches is that we can use read counts from high-throughput sequencing experiments to directly count the number of reads from each allele at heterozygous sites in the genome. So this basic framework can be applied essentially to any sequencing assay. And what we're going to do is measure departure from the expected 50-50 allelic ratio. So we can therefore measure allele-specific expression, allele-specific transcription factor binding, chromatin accessibility, and so on. And then when we measure these allelic effects in cells that are exposed to an in vitro treatment, we can effectively identify this genotype by environment interaction by testing for a departure from this 50-50 allelic ratio that occurs only in one condition, or that is statistically different across different exposures. So as I said before, one of the major advantages of this approach is the ability to identify this genotype by environment interaction even in individual samples. But importantly, we're keeping the trans environment constant because the two alleles are actually compared, the allelic effects are actually compared in the same individual sample. So I want to conclude by mentioning what we can do with massively parallel reporter assays. So we can use the same framework, but essentially what we have here is a reporter gene assay where multiple putative regulatory sequences are tested in parallels in the order of hundreds of thousands or more. So the advantage here is that we don't have to rely on naturally occurring variation, but we can design a panel of variants that we're interested in, for example, based on genome-wide association studies results or other sources of information. And the other big advantage is that we can use a variety of different cell types, because at this point, we don't need to use primary cells, we can also use cell lines, and we can use cancer cells as well, because we don't have the problem of having to deal with haploidy, because we are actually inserting our construct, which are biallelic by design into the cells. And so there's, these are like some of the technical aspects that this assay allow us to circumvent, and ultimately what will allow us to do, and this is something I guess we will discuss later too, is being able to explore a much larger set of exposures in terms of dosages, in terms of timing, in terms of composite exposures, and so on.
That's great. Thank you. So next up is Justin Colosino.
Thanks, David. And thanks so much for the invitation to join this excellent workshop. I really appreciate this discussion and the opportunity to think about how we can really start to best integrate epidemiological and experimental approaches. And so in my 5 minutes, I want to talk about the utility of patient-derived cultures in these studies to conduct what we could consider, epidemiologically informed toxicology to really understand environmental risks for cancer. And so there's really been substantial improvements in tissue culture methods in the previous decade that have really increased the utility of these models to allow us to understand chemical impacts specifically on target tissues. And like we just heard from Matthew, methods have been developed for the culture of primary patient tissues really across the carcinogenic spectrum, from normal tissue to pre-carcinogenic lesions, all the way to cancer tissues. And methods have also been developed to generate organ-specific cultures like neurons or intestines or lungs or kidneys from induced pluripotent stem cells. And now through in-depth profiling with single cell methods, we can really start to deeply understand the cells which are present and, the cells which are not in these different culture systems. I think we're really poised now to greatly expand on the existing model systems in toxicology to develop models which better reflect human diversity, particularly focusing on people who have been historically excluded from scientific research. And so I'll briefly talk about some of our group's experience in this space. So NIEHS and NCI graciously supported some of our work to understand environmental risk factors for diseases like breast cancer. And so we often use patient-derived normal cell cultures isolated from diverse donors in these studies, on which we performed multi-omics profiling at baseline. As an example, one of our projects is focused on understanding the health impacts of chemicals to which Black women in the US are disproportionately exposed, especially in the context of aggressive breast cancer. So these chemicals include a lot of the ones we've heard about over the last couple of days, pesticides, heavy metals, PFAS chemicals commonly found in personal care products, which can act as endocrine disruptors. There we can look at human biomonitoring data to identify the chemicals with disproportionate exposures. And then we can assay human relevant doses of these chemicals in vitro using robust dose response approaches. We're trying to optimize cost-effective and highly automatable, but also really data-rich methods, like high-content imaging based morphometric profiling with cell painting, which I guess in the context of this workshop would be considered phenomics, or things like high throughput transcriptomics. We can then start to define the biological alterations which occur at a given dose for these chemicals using benchmark dose response modeling. And then we can link these dose-dependent changes back to human exposure data to see which biological processes are changing at these human relevant doses. And we can conceptualize sort of like validation of these different findings in different ways. It could be intermediate biomarker associations in epidemiologic studies. It could be identification of some of these molecular exposure signatures, specifically in diseased tissues, or it could be looking at relationships between exposures and cancer-associated outcomes and secondary data sources. Or, we can start our own epidemiologic studies, like our new Michigan Cancer and Research on the Environment cohort study, which is part of the SECRET consortium. In general, I think this has been a really fruitful approach, but one of the challenges that we're seeing is that there's what I would consider really profound inter-individual variation in how cells from different people respond to a different environmental stressor at a given dose, including non-monotonicity in dose response. And so whether these are linked to inter-individual genetics, like Francesco was just telling us about, or epigenetics, or something else entirely, is really currently up in the air, but I think it's something that we really need to figure out because I think it's going to be really important. We also, of course, have the challenge in accurately modeling life course exposures and really complex exposure mixtures. And so I'm really looking forward to this continued discussion in the context of this panel to think about how can we best link these experimental approaches with epidemiological studies to help improve public health.
That was excellent. Thank you. Yeah. So we have time to start discussing some of these questions, and I'm just going to pose a few of them that were sort of we had before we came together, but we can take this any direction that we want to, because I think there are a large number of challenges that were discussed, and then lots of exciting opportunities as well in the future. And so, one of the first questions in formulating this session that we were thinking a lot about is, how different experimental models of environmental exposures can be used together with human multi-omics studies to enhance understanding of complex diseases. And then, beyond that, a little bit around what might be needed to facilitate this work in the future. I could see this by maybe starting with you, Francesca, because you mentioned these massively parallel reporter assays that are present, and we're seeing high throughput, high scale, massively parallel variant effect prediction assays. And so one of the thoughts there is we can generate multi-omics data, but maybe there's a way to go after specific variants in specific complex diseases. And I'm wondering what you see are challenges or limitations with this current state of the field.
Yeah, sure. I'll try to answer that question. It's a very broad one, I think, for like a few fields, right, from human genetics to toxicology and so on. So certainly, massively parallel reporter assays, or MPRAs, if you want to save some time, the name's pretty long, are one of the most promising approaches. And so the idea there being, I think that's what Stephen is hinting at, is that we can take the results from genome-wide association studies and essentially come up with a set of variables or even loci in the genome, right? Not necessarily individual variable, but multiple from each GWAS hit, and then test them. And so the question, I think, is what are we testing, right? And so we're testing the regulatory function of this variant in a experimental system which is simplified, like many of the systems that we've been going over in the past few days. But still, it allows us to do two things. One is achieve what's called fine mapping, right? So once we have a GWAS locus, we want to know which one of those many variants that are in disequilibrium is most likely to be actually the causal one for the trait that we're looking at. And importantly, what we can do is that, because, as I was mentioning before, because we're doing it in generally in a cell line and in vitro, we can expose these cells to stimuli. So we've been trying to do this in our group, and we've done. We have a preprint out where we've actually treated endothelial cells with caffeine, and we are now working on additional compounds actually funded by NIHS to do this work. And so the challenges are the same as, whenever we try to do an in vitro exposure to kind of replicate or simulate an in vivo exposure. So, timing of exposure, dosage of exposure, do we do one exposure, do we do multiple exposures? So certainly that. And then then we have the results, and we're going to compare these allelic effects, these genetic effects between exposed cells and non-exposed cells. And we will find some really strong effects, I can tell you, but we might not necessarily replicate or be able to validate all the signals that we expected to replicate or validate from the genome-wide association studies. And so the question there could be, is it because we're not looking at the right cell type, right? Because you need to be able to pick specific cell type system. And sometimes it's obvious, and other times the GWAS signal is actually the consequence of interaction between different cell types or a complexity of the organism. And then it's essentially episomal, so we're adding these DNA sequences in, but the chromatin context is not replicated. So what I'm trying to get at is that we're going to have a lot of false negatives. And so that's where then, I think what the field might want to do is build as much annotation as possible of putative regulatory variants in the genome in response to different exposures and in different cell types, but also, keeping in mind that the environment that we're replicating within the cells is not exactly the same that we have when we're looking at, you know, the native DNA and the native chromatin.
Yeah, no, I mean, that makes a lot of sense, and you know, probably touches on, you know, some of the topics that David was also suggesting too. If I'm just gonna spitball to you there about, you know, how you actually connect what's happening in some of the preclinical models to, you know, events that you're seeing, you know, in humans with this multi-omics data, you know, and I guess it actually touches on Matthew's talk as well, you know, where you're seeing like there's some evidence from the GWAS of things that might be present, but you know, how reflective of that is that from your earlier system?
Yeah, I'll offer that I think part of that is right. Like the endpoints don't always have to be the same, right? You know, they can be, you know, I threw out behavior as a sentinel endpoint to make connections between things that certainly, you know, to use the example of, I was talking about zebrafish studies, there's many parts that we don't have, you know, that zebrafish have, of course. So we're talking about different endpoints already. I think behavior is a sentinel. I think the other one is what, you know, Dr. Fry talked about with development as an outcome that offers a lot in terms of we don't need to wait for something long-lived to develop in it. I'm going to say elderly person, but even before that, right? Like it doesn't have to be an apical endpoint to matter in a developmental sense, right? You get a really, really early signal of that. So I think whether we're going back to organoid or in vivo models that have rapid development, you get that signal, or you know, birth cohorts, you know, things like that. Some of these early signals I think are a really great translational piece where it won't be a one-to-one perhaps mapping to the exact disease you're looking for, but certainly sentinels of those things I think are, you know, I'm going to say predictive, and hope that that's useful there.
Yeah, no, and actually I was thinking the same thing, you know, asking Rebecca too, based on, you know, her intro as well, is that, you know, we can see there's been a lot of interest in, you know, molecular endpoints, but then also these sort of clinical or endophenotype endpoints, or even cellular endpoints for trying to understand, you know, what are the impacts of environmental exposures. And so, Rebecca, I was interested in you know, given all the studies that you're doing with different exposures in the placenta, you know, where are you seeing like the biggest impact? Is it in that omics data, or are you really looking for those cellular endpoints that inform, you know, downstream interesting epidemiology?
Yeah, in some ways both. And sort of coming back to the initial question, one of the things that goes through my mind is that, you know, a way that experimental models can be used coupled with human population studies is in, you know, identifying mechanisms of action in terms of responsivity to chemicals of interest. So of course, with human population data, you know, we have confounding by lots of different variables like disease state or co-occurring chemical exposure or subject age. And even though that there are methods to deal with that, you know, the impact of those confounding variables is lessened in the experimental models. So I find, you know, one of the ways to, you know, no system is perfect. So having complementary approaches to try to get at mechanism is really important. Using the experimental models in concert with the data, the human population data, where you can really validate some target genes as biomarkers of exposure or effect by knocking them down or overexpressing them. And then once you have developed those, you know, those variants of the experimental models, those can be tested for susceptibility. I mean, some of where we're seeing really strong signals has to do with sort of placental cell biology and cell function, that many of the genes that are coming up as very important for human population-based research are the same genes that influence, let's say, cell growth and invasion. And we know that growth and invasion is so critical for a proper placenta, properly functioning placenta. So.
Yeah, no, that's great. Well, you know, it touches as well that, you know, you see convergence of genes and pathways, but I guess there was also some discussion earlier about convergence of different exposures as well. And, maybe I'll push this over to Justin to respond. How do you actually go after the entirety of different, exposures that might be present there? And then, is there information we can get from multi-omics data about the convergence of particular signals so that maybe we can actually, triangulate, important exposures instead of trying to test them all?
Yeah, I mean, I think that's a great question, very challenging question. So thinking about first, sort of quantifying human exposure, right, at the right time point is key to think about what are the exposures you want to look at. And so we've heard a lot about how we could do this on a modeling front or on a metabolomics front, mass spec, using these different technologies. And we heard some interesting discussion earlier today about whether we need to be prioritizing a set of exposures on the exposure assessment side to be moving forward. So I think conceptualizing that will be helpful for us experimentalists, because then we can take these exposures, if it's a smallish number in the hundreds, we can certainly assay these one at a time in like dose response modeling. We've got fancy robots that can help us pipette things, and really great microscopes that can take a lot of pictures and good sequencing data, and then that can help us to really correlate responses to different chemicals. But as we are trying to take some of these next steps with respect to exposure mixtures, I think that's where we're going to really need to brainstorm as a field. I think, we can take well-constructed mixtures. There's been some really beautiful work done looking at sort of like reconstruction of fracking chemical mixtures, for instance, and trying to understand the impacts that set of exposures has. And you can see this, some very potent effects on some cancer-associated phenotypes. What I think we need to brainstorm as a field is, do we need to be able to deconvolute the effects of the individual exposures and all potential mixtures within that exposure? Because then that just becomes completely untenable. If we want to look at like 30 different chemicals, there's 30 factorial potential mixtures in there for us to look at. So that becomes challenging. But to get back to what David was saying with thinking about these like next steps in sort of like predictive and computational toxicology, this is where the real power is going to be. If we can start to train these models to predict effects of a chemical, based on various chemical properties, see how good they are, maybe they'll be just good enough for us to then predict the effects of exposure in different organs. I have a lot of hope that in silico approaches are going to be evolving in parallel with a lot of the cool in vivo and epidemiological approaches to really move this field forward in that regard.
I think this touches on something that Alexandra was asking in the chat too, is that, not only do you have all these different exposures that you potentially have to mine, but then there's all these different contexts where you have to think about in terms of timing, whether or not it's an acute exposure or a chronic exposure, what the dose is as well. So I don't know how each of you thinks about that in your research. Are you going to the primary literature? Are you just, are you trying to just elicit a response in pilot studies? Maybe if one of you wants to comment on that. I'll send it to Matthew there, hand up, perfect.
Sure. So I think some of it's a mixture of the two. With regards to just piloting it and seeing what fits, that's sometimes what you have to do when there's just not a great deal of available literature. But also working with epidemiological experts in these kind of fields, they can really help you kind of narrow down what factors of interest you want to look for. Leveraging information from previous existing studies, we were really interested in the effects of ethanol, for example, on colon epithelium. And there's been a number of studies in other model systems away from the colon epithelium where they identify differences in stem cells and trans-amplifying cells. There are actually differences in mismatch repair that are, one increases it, one decreases it, for example, and trying to leverage that information is pretty challenging when you build these systems. With regards to kind of how you would get the timing for different omic layers, I think DNA methylation is a really interesting topic, for example, but it takes a pretty robust exposure to change the methylome. It also takes a little bit of time, and it requires the cell population of interest to be amenable to that change as well. Like trans-amplifying cells, they divide every 12 hours. So, they're pretty readily available to change their methylome to a very, very specific exposure. Whereas some of these, more differentiated cell populations are probably less amenable to that. So it's a pretty complicated picture, I think.
And it probably touches on David's point too about, you don't need to sample everything all the time. You'll learn different aspects from these different, omics modalities. I see Francesca has her hand up.
Yes, I agree with what's been said already. What I wanted to add, which I think kind of follow up on what you're saying, is that the way we approach it, and that's especially when we think about time of exposure, is to kind of keep in mind what's the specific scientific question we're asking. So for the studies that we're doing, we're specifically interested in this genotype environment interaction on gene regulation, how that translates to the complex trait. And so if we think about cis-QTL or cis regulatory variants, most of the times you want to look at like kind of early time of exposures, because that's when you have those direct effects of changing gene regulation, and you don't see a lot of trans effect, or you don't expect a lot of trans effect that you might expect later with a longer exposure. And so it really depends on what Matthew was saying before. If you're looking at methylation, you have to expose for a longer time, because the biological process you're looking at, it's going to take longer to manifest, right? So I think there's to learn from, like, it's clearly multifaceted, and ideally we like, to look at multiple times, multiple doses. But I think if we narrow our focus, or like keep the focus, not narrow, but keep the focus on the scientific question, then those technical aspects are kind of automatically implied many times by the specific question they were trying to ask.
Justin, did you want to add?
Sure. I think the two previous panelists really like hit the nail on the head, but really thinking about scientific question, like Francesca was just saying, is really important. I really liked how Matthew was conceptualizing sort of like cellular biology as reflective of like the omics layers you can measure. So yeah, so if we're just like screening new chemicals, we have like a 48-hour assay that we can look at like changes in cellular morphology, changes in RNA expression, that like works really well. But if you're trying to understand sort of like alterations in cellular differentiation, for instance, as like a predictor of carcinogenicity, that has to be a longer assay that would otherwise induce some sort of change in differentiation. Then you can look for like aberration in that trajectory. So I think really thinking about how you're pairing up your assay with the biology with the research question is really important, like the other panelists were saying.
Yeah. David, do you want to add?
Yeah, just to bring the NCI, half of the workshop, to the fore here. I think the question from Alexandra is a good one, but it doesn't even touch on like subchronic, right? So I mean, there's all these other kinds of studies that are important. So I just think it illustrates the importance of understanding these, whether it's a molecular initiating event or a mechanism of action or whatever your field prefers to think of these things as. I think that's really going to determine what you need. I think, Dr. Hill's presentation started with a mechanism from 1947, if I recall. I forget when that was, but I mean, there's a lot known already, and it seems to me that should actually guide the exposure timing more than the discovery goal.
Speaking of William's talk, one thing that I thought was pretty interesting with respect to it from a high level was not only do you have an experimental system, but you were actually reaching out to collaborators which had human systems and human data that was available there as well. And I'm wondering how much of a barrier do each of you have to finding, these types of partners where you can bring together not only exceptional models, but you've got the epidemiologists or the human data sets, particularly with these large scale population biobanks that are present there, and data across a range of models to sort of create a holistic picture of any of these types of studies. And maybe William, you want to comment on that, but I'd be happy for others as well.
Yeah, I'm happy to start. I mean, it was a lot of networking, a lot of collaborating. It's just a constant almost presentation of our data. This is what we're working on. Please come and work with us, come and talk to us, from a real wide range of fields, and from going to a simple cancer biologist to doing this kind of understanding epidemiology and getting to grips with the genetics and all the intricacies of setting up these clinical cohorts is huge. It's really difficult to try and establish these clinical cohorts and get all the clinical annotation that's required to really understand these cohorts.
Yeah, I was going to answer that from my perspective too, which is I feel really grateful to have collaborations with, for example, cohorts that started 20 years ago, right, to study developmental or placental origins of disease. That's not something you can really start today and look at tomorrow. So having, partnering with groups that do have those established cohorts and bank samples where we can look at chemical exposure, but also have some target tissue that's available. It's not easy, and I think it does, it speaks to team science and the importance of doing team science now, because again, no model is perfect. So having the samples that are derived from large enough cohorts with chemical exposure, cohorts with outcome data, and then ideally outcome data across a lifespan trajectory to help us understand how that changes. Not easy, but extremely important and valuable.
All right, so it looks like we've hit time. This went by really quickly. I'm sure we could probably actually spend much more time just chatting together about these topics, and hope we do get the chance in the future. Just to summarize, this session has really been about trying to expose people to different models that may be useful for studying gene by environment interactions and environmental effects and how they relate to disease, and more specifically cancer. We've looked at a range of different systems. There are a lot more that are out there and a lot of challenges that I'm sure people can see that are faced in terms of how to go after different exposures, choosing the right types of endpoints, whether or not they're molecular or not. And then also, as we touched on the end, building the types of networks that might allow people to advance their biology. So I just want to thank everyone for their time here this afternoon, and look forward to the rest of the discussion. So thank you.
All right, thank you, everybody. That was a great session. We're going to take just a really quick stretch break, and we're going to return at 3:30. Please come back for the very last session.